everyone. I'm Matt Fuller. I'm the head of gene therapy research at Ultragenyx and a member of the Patient Advocacy Committee for ASGCT. Developing treatments for rare diseases comes with unique challenges: small patient populations, limited data, and ethical questions about traditional clinical designs. Today, we're going to explore two tools that are helping researchers and regulators navigate those challenges: natural history studies and external controls, and how they're shaping the path to approval for gene therapies. Today, I am joined by Sam Hughes, who is a research program manager at St. Jude Children's Hospital. Sam, can you start by explaining what a natural history study is and why it's so important for rare diseases and gene therapies?
Sure, Matt, and thanks for having me today. Um, I think the first thing to understand about natural history studies is not only what they are, but what they aren't. It's important to understand that a natural history study is not a clinical trial. It's not a study where we're testing a drug or any other kind of intervention. What it really is a study where we are observing again the natural history, what we call the natural history, the normal, quote, normal progression of a disease over time, and ideally across the kind of age range of a disease, across the types of the forms of the disorder, if there's more severe or less severe forms, to understand what it looks like over time, what the progression is, meaning do people get worse over time? Do they stabilize? Is it fluctuating? And really, a main goal, objective of most natural history studies is to identify what are kind of quantitative outcome measures or things that we can measure in patients to really show how their disease is progressing, and how to ideally measure the potential impact of a future intervention, a future drug. So I live and work in the neurological disease space, and so we look a lot at these disorders in the context of neurological examinations, working with physical therapy and neuropsychology, and their tests that really break down the different aspects of the disorder. And it's especially important when the disease in question is very heterogeneous, or it looks different across different patients, or where everybody doesn't look exactly the same, and we need to understand certain parts of the population. So it's really trying to break down the clinical characteristics of a specific disorder. And in the context of gene and cell therapy, it tends to be driven by individual patients, the gene of interest that kind of puts the patients into a specific category, and also trying to understand how patients' different genetic makeup, even within one disorder, might impact the clinical picture. And so normally natural history studies can last for a long, long time, and try to collect as many patients as possible with the disorder. Some are for a very finite, a specific period of time. Maybe 5 years is a kind of an expected time frame to follow individual patients in a natural history study. But as you can probably imagine, it's very important in rare disease to be able to understand the progression of a disorder in as many patients as is feasible to track and follow over time. Because often when you have a very common disorder, you don't have to, you can make a lot of inferences. You can make a lot of guesses about what the general patient looks like, by following 100 patients or so, and getting like bigger numbers and understanding what the population might look like. But in rare diseases, especially ultra rare diseases, where there might only be 30, 50 or so known patients, it's really important to engage the patient community, not only to understand, for researchers and scientists to understand, like I was saying, what the patients look like over time, but to bring kind of the community together that would potentially be eligible for a future clinical trial of a gene or cell therapy. So it also serves a very practical purpose of understanding who are the patients around the world or in a specific nation, where they are physically located, so that we can really plan out a feasible clinical trial to really engage the active patients who can to ideally make it a successful trial.
Yeah, I think you make a really good point that we have to think creatively here, and we cannot take the templates from more common diseases and try to put them on rare or even ultra rare diseases. So I mentioned that there were two tools that we were going to discuss today, the second being external controls. Can you explain what it means by saying external controls, and how are external controls used in gene therapy trials?
The gold standard, what you always hear, is a randomized controlled clinical trial. And so I'm going to focus on the control, the C, the control part of that. So normally, if you have an intervention, a drug of any kind of intervention, you would enroll patients into a trial, and some would go on have the drug, and some would not for some period of time, maybe a year, maybe longer. Um, and normally that could be a placebo, so meaning not getting anything. Sometimes it's a comparator to some other standard therapy, an already approved therapy, and you're trying to compare the new therapy to a standard therapy. But it's all inside. It's inside of the trial. So meaning that everybody who enters the trial meets the same eligibility criteria. They look the same, quote unquote. They're all in the same age range. They all have the same kind of clinical features. So it's a way that getting to the point that we were talking about earlier about a more some populations are very heterogeneous. People look different even with the same disease. One of the points of a traditional clinical trial is to make the eligibility very homogeneous, meaning everybody looks roughly the same. And some people get drug and some people don't. And then you compare those and how they did over the course of that year or 2 years on how the placebo arm or the control arm either, followed the natural history of the disease versus the intervention arm that did better or worse or however they compared. Um, so that's really an internal control, what is traditional and what most people expect. When we talk about an external control, what that really means is that for whatever reason, and very often in gene and cell therapies, you see this where it's not really ethical or even practically operationally feasible to do this kind of randomization inside with the placebo. And often that's driven by kind of the progression of the disease, how devastating some of these diseases are, or just like the how we track and follow a gene therapy, and it can be very easy to find out from the researchers, from the study team's perspective, who got the gene therapy and who didn't. So how do we find a control? How do we prove to the FDA or other regulators, to the scientific community in general, that our intervention, our gene therapy, was actually changed the natural history of the disease? So we look at what we call external controls. We look at these, try to find these data sets that are often created through natural history studies, where we take, 20, 30, 40 plus patients that have been followed without intervention, um, who that theoretically represent the progression of disease in a given population, and we compare that historical data set to the data set of treated patients in a clinical trial, however many that may be. And so the idea is that you don't have to have this concurrent control group along with a treatment group, meaning happening at the same time, following at the same time. But if you're able to take a data set of an appropriate number of patients that had the same kind of assessments done, especially like how they followed the disease, or they're the same kind of PT assessments, or the same kind of psychological assessments, or whatever kinds of testing were done in the natural history study data set, compared to the treatment data set. And can we as researchers parse out, did the gene therapy or cell therapy actually make the change the trajectory of the disease course in the treatment group than what we saw in a historical natural history data set?
Thanks. That makes a lot of sense. And I think what you're getting to is utilizing these tools to support approving a therapy. Can you explain, how a natural history study with or without an external control, can support getting a therapy approved?
Right. It's really been in the last 10 years or so that it's that the concept of external controls and natural history studies as a way to get an external control data set has been really more thought about and considered by the FDA specifically and other regulators. You can have a natural history study, and within a natural history study of, say, you're following 50 patients over time, it might not be that entire 50 patients that is the external control data set. It actually might be a subset of that study that will be the external control data set. And that's driven by factors like what part of the natural history study participants actually looked like the patients that were in the clinical trial. So it might only be like 20 patients of the 50 patients in the natural history study that are comparable to the population that's in the treatment trial. It's the matching control to the patients who are enrolled in the clinical trial of whatever gene or cell therapy. Because that's the key, is that they have to look alike to be able to compare to each other. And sometimes not the whole natural history study is fully comparable to the treatment population for any number of reasons. And so that's one of the considerations that researchers have to take in, not only when they're designing natural history studies, but also when we are looking at historical data and saying, can we use this in drug development?
So, Sam, I'm hearing that there are a lot of options and a lot of things to consider, not surprisingly. How can patients and patient advocacy groups understand what health authority regulators like the US FDA are looking for? How can they put together a package that will be successful?
The first place to go is the guidance. There's a 2019 guidance that was presented from the FDA around the use of external controls in rare disease. And that's kind of the foundation. And that actually was driven, they created that guidance based on researchers and scientists that had been doing clinical trials and were proposing the use of external controls. That proposing these uses, and the FDA was saying, yeah, it works. We can actually make this work, at least in specific kinds of disorders. And kind of the hallmark kind of disease that we point back to, that's really the most exemplary of on this topic, that has has had the most impact to really making that guidance, was the approval of Zolgensma, the gene therapy for spinal muscular atrophy, or called SMA. That's kind of the gold standard here, where it was the kind of the perfect constellation of a very severe disease that was frankly progressive, than the most severe form of SMA, which is about 60% of the population, of the SMA population. And that the outcomes in SMA are time to death or time to permanent ventilation, which are very like survival oriented outcome measures, as opposed to something that's really intricate, like a psychological outcome measure, some kind of developmental measure, that kind of thing. Um, and so they were able to really take that outcome measure, that patient population, and what was known about the disease, and the historical work that had been done through two separate observational natural history studies from two different groups, um, to say, hey, we did this open label, meaning everybody in the trial got the gene therapy. Take that treated population and compare it to these two natural history studies that both showed that there was that it was completely different, that the gene therapy had completely transformed the trajectory of the disease. But it's been most effective, most impactful at the FDA level when it's been a more frankly degenerative disease, when there's been more survival related outcomes that are primarily what we're following in the disorder. And it's been less successful or more complex in making an argument with the FDA when it's been more slowly progressive disorder, when it's been more developmental or behavioral based diseases.
Thanks. We've heard a lot about, the role of researchers, the role of clinicians, executing these and designing these. But we also know that patients with rare disease and their caregivers know more about these diseases than clinicians because they live with it every
Right.
day. Can you elaborate on what role patient advocacy groups, families, patients with rare disease, and caregivers play in building these types of studies?
Yeah. So really they're the driving force from the beginning. And I would say often, if not the vast majority of the time, natural history studies are driven initially by some kind of advocacy group, big or small, in the space. Often working with clinical sites, with researchers. But it can start even as seemingly small as just as a contact registry of patients to understand again, like I was saying before, where the patients are. In the case of genetic disorders, if they're willing to share a genetic report, what is the genotype? What is what do the patients look like from a genetic standpoint? And kind of that's a starting point of a natural history study. Sharing surveys that the patient advocacy groups might send out about what matters to the patients and the families. Kind of trying to corral medical records that people have. These are all things that are normally part of a natural history study that like an academic site would do. So honestly, the more that an advocacy group can again kind of corral the community and get this information just from standard medical records and be able to look just retrospectively look backward at how people looked over time, that's a great starting point to help the researchers and the clinicians think critically about the design of a natural history study, of a more formal perspective, what we call perspective longitudinal, meaning forward looking and connecting or collecting multiple time points over a period of time, and in a standardized way. There's always often advocacy groups or foundations fund this work initially. It's there are times and there are funding mechanisms through the federal government that can support natural history studies that even advocacy groups can apply for themselves. It's vital to be able to support these patients as much as possible to participate in the trial, whether it be coming to a site or some other kind of virtual way that we can engage with patients in a way that is still rigorous and allows us to collect appropriate data. Finding and identifying what we call the patient voice, and getting that in early when we're designing these studies and not design a study and then ask, what do you think about it? Advocates, patient representatives, and them saying, nobody's ever going to enroll in the study because it's not feasible or it's too much effort, it's too much this, too much that. So it's very important to engage these patients early on, and the advocacy groups can really take the lead in bringing the voice to the table.
So what you've described to us so far is really sort of the cutting edge of what's happening with natural history studies and external controls. If you look ahead, how do you think the use of natural history studies is evolving in rare disease research?
It's become like we who've worked in the rare disease world for a long time have been shouting this from the rooftops for a
Yeah.
while. Like the current structure of randomized controlled clinical trials don't work for rare diseases, and it's seeming like it's finally starting to hit at the highest levels.
Yeah.
So it really makes this kind of this historical information or concurrent observational studies of natural history studies or whatever we want to call them, that much more important in the drug development process. Historically, I would say natural history studies were just a thing that people did, looking forward to some future, maybe something will happen. But like I was alluding to before, we're actively in drug development while we're designing and doing a natural history study for these disorders. And so we have, in some ways, that's very daunting because you're like making the plane while you're flying it. But it also gives us an opportunity to mitigate some of these issues that have been difficulties for researchers who have been trying to use old historical natural history data sets that were not fit for purpose for drug development. But now we can make these natural history studies that are truly fit for purpose in this drug development, specifically gene and cell therapy. Um, and so being able to understand at a population level, even a small population level, what that looks like, how what the clinical picture of the disease is, and even at the individual patient level to say, I'm making a personalized therapy for this particular individual or individuals like this patient, and we need to follow them for 1, 2 years in advance. That's a natural history study just at the end of one level, at a single patient level, to know what they look like to compare them to themselves. So that's there people are getting the FDA and other leaders in the space are getting are open to a lot more of this kind of creative thinking, creative designs and structures around rare disease drug development. And so we have this opportunity as the researchers in the space to capitalize on it and do our own due diligence to do it well, do it properly, and propose ways that we think that it can be better for now and in the future.
Dan, thank you so much for your time and for sharing your expertise. We hope this was informative and useful to the community.
Hey, thank you, Matt. I appreciate the time.